Okay, we're going to start talking about antibodies now, the humoral immune system. Let's review. The humoral immune system is involved in fighting pathogens that are extracellular, outside the cell. B cells, known as B lymphocytes, secrete antibodies. Antibodies can go by the name of immunoglobulins or gamma globulins. So it's three names: antibodies, immunoglobulins, or gamma globulins. They're secreted by B cells. And remember, I mentioned, once fighting B cells start secreting, when they get to 2,000 secretions per second, they're called plasma cells. So plasma cells are B cells in hyperdrive secreting antibodies. They're capable of binding specifically with an antigen, and so we go further. The basic structure of an antibody is this here. Okay. You have two heavy chains. Now this would be in the quaternary structure of proteins. You have two heavy chains. They're longer, have more amino acids, and two light chains here. They're hooked together by what we call disulfide bridges. This area up here is the variable end. That's where the antigen binds. Actually, this tip is called the hypervariable, and back here is the variable. Back here is your constant region. These are your constant region. Listen to me well. The amino acid sequence of the constant region determines the class of the antibody. The amino acid sequence of the constant region determines the class of the antibody. There are 5 different classes of antibodies. There are 5 different classes of antibodies, and they all do slightly different as far as how they work. The 5 different classes of antibodies, and we'll come back in a moment, is IGM. Now the IG stands for immunoglobulin. An IgM, an IgA, an IgD, an IgG, and an IgE. Okay, so there are 5 different classes. 5 different classes of antibodies, and we're going to go through that in just a second here. But I go back, this is the structure of an antibody right here. All right. Here are the different classes: IgM, IgA, D, G, and E. Okay. Now, this is what I want you to remember before I go back to that. How do antibodies do their job? And then we're going to come back. In other words, this is what we've laid out. There are 5 different classes of antibodies. You have a constant region and a variable region. And at the hypervariable region tip, that's where the antigen binds. The constant region, the amino acid constant region, determines the class of antibody. That means the constant region's amino acid sequence of M is different than G. Okay? Now I go to when an antibody attacks, what does it do? How does it really get rid of the organism? Okay, here's what I want you to remember. PLAN, plan. PLAN, plan. That's a mnemonic. The P stands for precipitation. It can cause precipitation of the foreign substance. Precipitation means to rain out. It makes it heavy because it can join to a bunch of them and make it heavy and drop it out through the tissue fluids. The L stands for lysis. The L stands for lysis. The lysis occurs through the use of complement. Remember the complement system we talked about. The A stands for agglutination. Agglutination means to clump together. It sweeps them up in a pile. I want you to read on this, by cross-linking. And the N is neutralization. Because remember, in a microorganism, we said that the end that causes the trouble is the tip, those epitopes. And if you come in and the antibody would cap over the tip, if it caps over the tip, then it essentially would neutralize that. The organism would still be there, but it would neutralize. So plan, P-L-A-N, precipitation, lysis, agglutination, and neutralization. The lysis occurs because, again, of the complement system. Go back and study the complement system and the membrane attack complex. Remember I told you please go back and check that in your book or that monomer IgM or IgG can bind complement, and once complement is bound, it can cause several things to occur: opsonization, inflammation, and lysis. Okay, so we go back again. Let's now look at the classes of antibodies. Now, a B cell can only the specificity of the antibodies of B cell is only for one type of antigen. That means if it's Streptococcus A, it will only do that. However, so the B cell cannot change the specificity of the antibody for the organism. So then what's the different classes about? The different classes depends on where the organism is. So let's say like this. Let's say theoretically that we created a bullet that had a backing on it and a tip. The tip we made to target someone. Now, the person's trying to get away from the bullet, and if you shoot the bullet, it could go all around the world, let's say, and find that person. Now, the backing of the bullet would depend on where it can go. If it needs to go up in the air, you'd have a different backing, same tip. If it needs to go into oceans, different backing, same tip. If it needs to go through a mountain, different backing, same tip. So you're changing the constant part, but you're leaving the tip the same because it's still targeting the same entity. It's still so when we look at it, let's go back here. Okay. Here's IgM. IgM can exist in a pentamer form, 1, 2, 3, 4, 5 in a ring, or monomer. The monomer form you don't see here. IgM is the most archaic antibody, evolutionarily. It's also the first antibody to be produced in an infection that stimulates the B cells. So, if I'm looking for antibodies to see if you have an infection, sometimes we can't find the organism. We have to look for the antibodies, and where there's smoke, there's fire. They'll say that you have this infection. So, if I look and the only antibody I see present is IgM, then I know it's a very recent. So you can see doing a primary response. Read on this. That's IgM. IgA is one that dissolves in secretions. It has this secretory piece right here. And this secretory piece allows it to dissolve in fluids. Now what it tries to do let's say it gets in your bladder. Let's just use that. Then what it would try to do is coat the epithelial surface in your bladder to prevent the microorganism from attaching there. Okay, so we did IgM, IgA. Now you need to read on it too. Here's IgD. The only thing that IgD does, it never gets secreted. It's one of your surface antibodies. So IgD is one of your surface antibodies. Okay, so your surface antibodies actually are, let's go back here. Your surface antibody can, in some cases, be an Ig monomer M also. Okay, so IgD attaches to B cell surfaces. Okay. IgG is the most abundant antibody that you have. It's the one that generally is the second to come out, and it's the one that is present in secondary and late. It can also cross the placenta. Okay. IgG can cross the placenta. IGM cannot. And I'll bring that back up again at a time. Okay. IgE is kind of a pesky antibody. It's the one that most don't like to see. It's kind of the one that does inflammation. IgE attaches to mast cells and basophils and causes them to release histamine and heparin. So it's active in allergies and parasitic. So here's D, here's G, here's E. Okay? So those are your antibodies, and you need to read on that in the text also, whatever text you're using. Now remember I told you about the skeleton key deal, that your parents handed you some skeleton key fits to things that they had seen in their environment, it was in their genetics, and the genetics before them. They only hand you a few million, but you can make a billion. You can make billions of antibodies. Through this, we talked about somatic recombination, rearrangement of somatic genes, epigenetics. You can change your genetics after you live. Now, to give another example, identical twins born, move apart. One identical twin moves to India, another moves to England. They may not be the same. They may have different genetics slightly because of epigenetics, and they definitely will have different production of antibodies between the two countries that they are in. Antibodies inactivate and tag, that's the neutralization, agglutination. So what the antibodies do is attach to several antigens, and then several of them, it makes it real heavy clumps, and they fall out of solution. And when they fall out of solution, that's called precipitation. Now, the precipitation then allows macrophages and neutrophils to come and eat it. So neutralization blocks specific sites on vac and exotoxins, agglutination, precipitation. Here's the complement fixation. Remember, the complement fixation, as I alluded to, can trigger the lysis. Go back and study complement. So plan, precipitation, lysis, agglutination, and neutralization. Okay? We talked about IgE, that's the pesky one. Here's another drawing that you can look at on your own. Monoclonal antibodies basically are produced in this way. What you basically do is, in the laboratory, you create an antibody for something. This is in this biotechnology. Create a B cell. You create a B cell, cultivate it to produce antibodies towards something specific that you want it to be. Then what you do is you take a tumor cells. Now, tumor cells can multiply ad infinitum. So you hook the tumor cell up to attach it to the B cell. That's going to make them proliferate mitosis a lot. That's what we call a hybridoma. That way you can produce a lot of those specific antibodies. Now, you could have made antibodies against human chorionic gonadotrophin. That's a hormone produced in pregnancy. If you did that, then, as a female, you could urinate in a little tube. The antibodies would be in there. And then the antibodies, if there's any HCG, would attach to it, cause clumping, and you'd see that circle at the bottom of the test tube saying that you're pregnant. You can create antibodies against diseases. That's antibody therapy. That's a new way we're looking at to do for cancer cells. Produce antibodies against, call it immunotherapy. So this is what we call monoclonal, because they are one clone, monoclonal antibodies. We'll end here, and next we'll talk about cell-mediated immune responses.